Greetings and humble salutations. I am Rebecca Oyetoro, a third-year biology student all the way from the home of the Marching 100, Florida A&M University. This summer, I had the opportunity to work under the University of Florida SURF. I worked in the College of Agricultural and Life Sciences in the Department of Microbiology and Cell Science. I got the opportunity to work in Dr. Julie Maupin-Furlow's lab, and she was my designated faculty member for the summer. And over here is my PhD mentor, Sungmin Hwang. In this experiment, I was assigned the task of determining the effects of regulatory proteins in oxidative stress from Haloverax volcani. So, let's start with understanding what a Haloverax volcani is. Haloverax volcani is an archaea. They live in extreme saline waters. They can be isolated from oceanic environments with high sodium chloride concentrates. They are also chemioorganotrophic, which means they are primarily aerobic, yet they are capable of anaerobic respiration under anoxic conditions. Now, the focus of my experiment was to analyze the impact of oxidative stress, but it is important to know that oxidative stress is caused by reactive oxygen species, which are initiated by oxidants an organism is exposed to. Although oxygen is necessary to survive, too much oxygen can be very life-threatening, causing damage to your proteins and DNA, which may lead to tissue injury and inflammation. So, how did we come across the TRIM-B-like gene and the hyper-R gene? Well, due to prior studies from Dr. Maupin's lab, the TRIM-B-like gene and the hyper-R gene supposedly have an effect on oxidative stress. Now, back to the experiment. We would like to understand how oxidative stress is regulated when TRIM-B-like and hyper-R regulatory proteins are deleted. What are their functions? These are questions we hope to find answers to as we conduct our research. In order to begin, we had to create the most important part of the experiment, the Haloverax volcani, with the deleted genes, of course. Now, this process took us a month and a half. Crazy, I know, but it had to be done. In order to do this, we had to generate pre-chaoplasmid, generate chaoplasmid. We went through the pop-in phase, the pop-out phase, and then we had to confirm our knockout. Generating pre-chaoplasmid was vital in generating chaoplasmid. With these methods, we started from our computer desk, where we observed the size of the DNA and the target gene, which gave us an exact idea of what size to expect. To confirm the deletion, we sent the samples to get sequenced before we moved forward. The pop-in phase and the pop-out phase were a bit tricky. During the pop-in phase, our Pri-E2 plasmid was integrated into the genome. During the pop-out phase, we treated the media with 5-FOA, which made the Pri-E2 gene toxic. 5-FOA synthesizes the enzyme orthidine 5-phosphate decarboxylase, which limits cell growth. We expect two results, a wild type or deletion mutants. To determine which is which, we perform PCR, to allow us to determine the deletion mutants based on their sizes. My mentor and I worked closely this summer. With his assistance and direction, I was able to undertake the protocols easily. I got the chance to perfect my skills in inverse PCR and colony PCR. I got to streak bacterial culture, perform gel electrophoresis, and get an image of my gel using the UV transilluminator. Overall, I enjoyed my summer working in Dr. Maupin's lab. I learned the importance of being patient and consistent with my experiments. Also, I got the chance to improve on my wet lab skills. I plan to continue on a research track, and I would like to implement my research skills on more medical prospects.